Hi everyone, welcome to a Tess Academy YouTube video. This is part of our GCSE Biology revision lesson series. Today we'll run through mutations. Remember, if you have any queries, please visit our website at tessonlineschool.com. Also, please suggest any feedback, improvements or suggestions to us through the website. Thank you. Let's start off by defining mutations. Mutations are random changes which occur in the sequence of DNA bases. They are rare and random, and sometimes they can be inherited as it occurs in the DNA, which is passed on from the parent to its offspring. Mutations occur continuously, which means they are always being formed and can be spontaneous and random. Some of their causes include ionizing radiation or chemical mutagens such as tar from cigarette smoke. We will now go through the types of mutations, the first being insertions. As the name suggests, insertions occur when a new base pair is inserted into the DNA sequence. An insertion mutation changes the amino acid that would have been coded for by the group of 3 bases in which the mutation occurs as the base sequence is changed. This also produces a knock-on effect by altering the following groups of 3 bases on the DNA sequence. This can be seen in the diagram as a guanine is inserted in the second triplet, which changes this particular triplet and therefore codes for a different amino acid, i.e. we go from a serine to an arginine. The bases that follow after are shifted towards the right, which ultimately causes a frame shift throughout the whole DNA sequence. The next type of mutation we will look at is deletion. Once again, the name suggests that deletions occur when a base is randomly deleted from the DNA sequence. A deletion mutation changes the amino acid as the triplet code is changed. That would have been coded for by the group of 3 bases in which the mutation occurs. This also produces a knock-on effect by altering the following groups of 3 bases on the DNA sequence, consequently causing a frame shift. For example, the thymine in the second triplet is deleted, so all the bases shift one position to the left, causing a massive frame shift. Thirdly, we will go through substitution. Substitution occurs when a base in the DNA sequence is randomly swapped for a different base and therefore substituted. However, unlike an insertion or deletion, a substitution mutation will only change the amino acid for the group of 3 bases in which the substitution mutation took place. Therefore, it won't produce a knock-on effect. This is because there is no frame shift as the position of the triplet codes does not change. Let's see this in the diagram. For example, you can see in the diagram the thymine is substituted by the adenine and only changes the second triplet, but the first and third triplets stay the same as there is no frame shift. How can mutations affect an organism? We know that the DNA base sequence determines the sequence of amino acids, which then go on to make up a specific protein. Since mutations alter the DNA base sequence, they will change the sequence of amino acids, therefore the mutations will lead to a change in the protein. However, most mutations do not alter the protein due to the nature of DNA or the mutations themselves, or only slightly alter it, therefore its function and appearance remain the same. But some mutations do code for a significantly altered protein. This may affect the ability of the protein to perform its function. For example, if the shape of the active site on an enzyme changes, substrates won't be able to bind to the active site anymore. Structural proteins such as collagen may lose their strength if its shape completely changes. Let's put our knowledge of mutations that we have learned now to the test and answer the following questions. Try these questions independently and return back to the video to look for the answers and add any corrections. So let's first start off with this multiple choice question. The question is, the sequence of bases in a gene is as follows: A, T, T, C, G, G. A mutation caused a change in this sequence. The new base sequence is A, T, T, C, G, G. Which of the following took place? Is it A, repetition, B, deletion, C, insertion, or D, substitution? The answer is C, insertion. You can see that A, an adenine, has been inserted in the second place in the base sequence. The next question is also another multiple choice question, so let's have a go at this now. Which of the following is not a type of mutation? Is it A, repetition, B, deletion, C, insertion, or D, substitution? The answer is A, repetition. You have learned the following deletion, insertion, and substitution mutations above. Let's now try a longer question. This is a 4-marker, so it's integral you add as many details as possible to be able to get all the marks. The question is, compare the following mutations, insertion and substitution. Give yourself a mark for each of the following bullet points. Remember to add any corrections if you have forgotten to add any of this information. Firstly, insertion occurs when a new base pair is inserted into the DNA sequence. Then, substitution occurs when a base in the DNA sequence is randomly swapped for a different base. Then, insertion produces a knockoff effect, or it alters the following groups of 3 bases on the DNA sequence. Lastly, substitution only alters the amino acids for the group of 3 bases in which the mutation took place, or it won't produce a knockoff effect. Remember that you are comparing, so you have to use comparison points for both insertion and substitution. Thank you for watching. Remember, if you would like further support, we offer one-to-one sessions and group sessions for key stages 1, 2, and 3, along with specially formulated 11+, GCSE and A-level lessons. Further information can be accessed on our website at tessonlineschool.com. And if you would like further resources, advice, and feedback, please contact us on the following numbers. Thank you.